Hello, everyone. Today, we want to do a topic that I think many of us find very interesting, and we want to talk about emotions. And in talking about emotions, we want to talk about a series of subcortical structures known as the limbic system. So let's get right into it. Now, one of the persons who studied emotions for many, many years, Joseph Ledoux, made this statement. He said one of the most significant things ever said about emotion may be that everyone knows what it is until they're asked to define it. Now, that might seem a strange statement, but you think about it. If I asked you to define what is an emotion, what would you say? How would you describe it? We know it's a very important component of the human experience, but to pin down exactly what it is something that is a little bit more difficult. And it's something that scientists, psychologists, and neuroscientists have been exploring for many, many years. So here's a definition. One definition of an emotion says it's a complex psychological state that involves three distinct components that you can see in the image there. The first component of emotions is that of feelings. And I suspect all of you, in coming up with your definition, would have mentioned feelings. Feelings is the subjective experience that you experience. It's that subjective sense that you experience. It's not neutral. It's either positive or negative. And it's what we most often associate with emotions. But emotions also have another component. They have a physiological response. And that usually involves activation of the autonomic nervous system, especially the sympathetic nervous system. So examples of components of the emotional response that are physiological would be increase in heart rate, or increase in sweating, or dilation of blood vessels leading to flushing. And then finally, the third component is the behavioral or the expressive response. And this now leads to actual behaviors that may be linked to purpose and motivation. One of the reasons we believe we have emotions. This could be linked to facial expressions, which has connections with communication. Another reason we have emotions. So that third component is the behavioral or the expressive component. So taken together, these represent the three components of emotions: the subjective, the feelings, the physiological, and the behavioral or expressive. Now, persons have been thinking about these things for many, many years. And two scientists, James and Lange, put forward a theory over 100 years ago, which might seem a little strange as we explore it. But let's track and understand what they were saying. The James Lange theory of emotion can be summarized by this statement. We do not run from a bear because we are afraid. Rather, we are afraid because we run from the bear. Now, in that statement, afraid is the emotion. They're describing the emotion of fear. And what they were basically saying, if you look at the diagram there, is you see something that's potentially frightening. You see a bull rushing you, or you see a bear. And that produces a set of responses, one of which is running away. And when you begin to run away, you look at yourself, you almost internally look at yourself, and you notice you're running away, and you say, aha, the fact that I'm running away means I'm afraid. And that's how you label the emotion. And so what James and Lange were basically saying is that the labeling or the defining of emotions comes after the behavioral and the physiological responses. So there's a stimulus, then the physiological behavioral response next, and then there's the labeling of the emotion. Now, this might sound strange, but you can't just say something sounds strange. You have to be able to give evidence for why it might not be true. The most obvious reason why this might not be true is the fact that, well, if you think about emotions, if you think about the emotions of fear, about anger, and about joy and excitement, they almost all have similar physiological responses of increased heart rate, of increased pupillary dilation, of possibly sweating as well. In other words, there's not a unique physiological and behavioral response for each emotion. So if this theory were to be true, the question would then be asked, well, how do you know which emotion you're experiencing when you're running away from the bear? Is it anger? Is it fear? Is it excitement? Having said that, we should point out that in modern psychology, we do recognize that the physiological state of the body can actually affect emotional states. And this has been used in two things: the facial feedback hypothesis and also laughter therapy. So if I were just to ask you to smile now, or put a smiley emoji on your screen, or pretend to laugh, what you find is that if you keep that up for a few seconds, it actually changes your mood. And there are studies that have verified this. And there are also studies that have shown that this can be used for different types of treatment related to different psychological conditions. Then there are studies that have looked at the facial feedback hypothesis, where a famous study is people were asked to hold a pen in their mouth. Depending on how you held the pen, it would shape what expression you had. So if you held it with your lips, you would have a frown. And if you held it with your teeth, you would have a smile. And depending on how you held the pen would then affect your immediate emotional state. And just interestingly, looking at this facial feedback hypothesis, there has been work now looking at experiments with Botox. Well, not really experiments, but by studying persons who have taken Botox treatment. As you know, Botox is used to help get rid of wrinkles. And when someone has received Botox, they have difficulty frowning because the muscles of facial expression, some of them have been paralyzed. So you can see that there in the top diagram here. This is the person with Botox, and if they're asked to frown, they cannot because these muscles have been paralyzed when compared to the control. And when you look at the amygdala activity, now the amygdala is an important brain structure for processing emotion. You found that the persons who had Botox therapy had reduced activation inside of the amygdala. And some of them also report less negative moods. So these are all examples showing that maybe James and Lange weren't totally wrong, that the bodily state and your level of physical, physiological, expressive states within the body can actually feedback and influence your emotional state. Now, the second theory to come along after that of James Lange was the Cannon-Bard theory. And they basically said that when you perceive a stimulus, that perception occurs at a subcortical level. It was suggested the thalamus. And from the thalamus, two things happen at the same time. The thalamus helps, those subcortical structures help to trigger the physiological response. And at the same time, at a more cortical level, you have the subjective experience of emotion. And so Cannon-Bard were putting forward the idea that at the same time, you were getting the subjective feeling of emotion and the physiological arousal. A little different from James Lange, who put the physiological responses before that's cognitive labeling. This idea lasted for quite some time until the 1960s, when another two scientists put forward the idea of the two-factor model. And they spoke about the importance of context. And their work came out of a very simple experiment in which participants were given an injection of adrenaline or a placebo. Now, you know, if you are given adrenaline, it will produce many of the symptoms of sympathetic arousal that often are associated with emotional arousal, increased heart rate, increased breathing rate, sweating. Now, when these participants were given the injection of adrenaline, some were then put in a room where actors were told to behave in a certain way. Some were told to behave in an angry way. Some were told to behave in a happy way. The subjects who went into the room and they didn't know about the experiment and they just found themselves around happy or angry people, they found that when you were asked about their emotional experience, they said either they were happy if they were in the happy room or angry in the angry room. In other words, they were taking the interpretation of how they were feeling because of the adrenaline from their context. The persons who explained this is what the adrenaline is going to do to you, when they went into their room, they didn't take their interpretation from that. They knew the adrenaline was the cause, and therefore they didn't report a change in mood. And this led to the whole two-factor model, which says that in thinking about emotions, physiology plus context is important. So you will experience an emotion. Say you feel your heart racing. You want to, you feel your heart racing. You know you're starting to sweat. But the context in which that is occurring is important. So some people might see the bear, they might want to run away. And then they'll say, okay, I'm afraid. But other people might know about bears and they might understand bears and they might not think they need to run away. And they might be excited. They might say, oh, wow, look at that. And so the context is important to help label the emotion. And then having labeled it, it then leads to what is your feeling and what is your action. So this slide here summarizes what we've just been saying. The James Lange theory says you see the stimulus and it produces arousal. And once that arousal takes place, after that, you label the emotion. The Cannon-Bard theory says you see the stimulus and at the same time, you have the physiological arousal and the labeling of emotion. And then the Schachter-Singer two-factor theory says that you see the stimulus, you have the physiological arousal, but the context, what's the context? Is it late at night? Does it seem like somebody is breaking into your house? That context helps you label the emotion and give rise to the fact, well, yes, this is fear. All of this to say that emotions are quite challenging to understand. And it's important to have definitions and understanding if we're going to explore the underlying biology or neurobiology of anything. So let's ask another question. What are the functions of emotions? Well, we've already started to define these functions, even though this still is not totally clear. So some of the functions of emotions, it seems, is they're linked to motivation. So for example, when something dangerous is occurring around you, that emotion propels you to protect yourself by possibly running away or preparing to fight. The other things we do know associated with emotions is that most emotions can facilitate learning and memory. We know emotions also can affect sensory thresholds and therefore can make us more sensitive to particular stimuli. And we know emotions can also affect communication with others and therefore modulate approach and avoidance behavior, as in courting. And finally, we know that emotions can affect performance, and that therefore might be another important role of emotions. Again, difficult to pin down, but we do see that persons who have difficulty processing emotions and lack emotions, there are a lot of normal human behavioral functions that they find difficulty engaging in. Okay, so having given us a little bit of a history and understanding of what emotions are, why we have emotions, how scientists have sought to define emotions, we want to ask the question, well, what are the brain processes, or are there brain processes that are responsible for emotions? And to begin this part, we want to look at the work of a gentleman called Paul Ekman. Now, Paul Ekman started off his work trying to determine whether emotions were universally the same. His idea was perhaps they were not, and they were the result of socialization. So he traveled to many parts of the world to explore emotions in different cultures. And he was surprised to find that actually wherever he went, emotional expressions and emotions were pretty standard across different cultures. It was then suggested, well, maybe the reason this is so is because the world is interconnected, and everybody is experiencing relatively the same social constructs around them, whether you're in Japan, whether you're in the Caribbean, whether you're in the United States or Europe. And so Paul Ekman decided to go and spend some time in Papua New Guinea with a tribe that had not been exposed to modern society and study their emotional responses. And to do that, he would read statements to them and ask them how would they respond. And you can see 4 statements on the screen there that he read. Your child had died. You see a dead pig that has been lying there a long time. You are angry and about to fight. Your friend has come and you are happy. And then he would ask the persons to say, what would your emotion be? And he would take a picture, and you can see some of the examples there. And I'm just going to ask you to take a moment and match the statements to the examples. These are the answers. Well, I think the one that's most easy for all of us is, your friend has come and you are happy. That's A. I think that smile is universal. Just like your child has died, B. That's pretty universal as well. The other 2 might be a little bit more difficult, but I think when you look at them, you notice it. You see a dead pig that has been lying there a long time. And when you think about a dead pig, you think about stench, you think about smell, you think about something that you don't like. And you trigger the emotion of disgust. You screw up your nose. And that's D. And then C there is the issue of fighting, anger, getting ready to war. Now, based upon the fact that these expressions were exactly the same that you found in other cultures, Paul Ekman had to conclude that it seems that there are universal emotional expressions. Now, anytime something is universal, you can begin to think, well, that's not usually a result of the environment. That's probably the result of some underlying biology. And Ekman went on to describe 6 basic emotions: happy, sad, fear, anger, surprise, and disgust across a range of countries all over the world. And it's also been said that you can have these in babies. And I'll let you look at those images and link them to the 6 basic emotions described by Ekman. So the work of Ekman tells us that if everywhere all over the world human beings experience the same emotions regardless of culture, well, probably there's some underlying brain process controlling emotion. But let's look at some other work from clinical studies that might support this. Now, these are some rare conditions. Well, one of them is not that rare. Let's start with this lady on the bottom. Now, this lady has experienced a stroke, and the stroke has affected her left hemisphere. Now, we know the left hemisphere controls the right side of the body, exerts motor control on the right side of the body. So if you ask this lady to make a smile, make a false smile. Imagine you're a model and you're walking on that catwalk and you have to make a false smile. What happens is she will try to smile, right? She will try to smile. And you will notice that the left side of her face responds because that's being controlled by the right side of her brain. But the right side of her face does not respond because she has damage to the left side. And that's consistent with the stroke that she has. And that's something that we see in many, many stroke patients. Remember, don't get confused looking at these images with right and left. Because they're pictures, remember there's lateral inversion. You have to turn yourself around to work out what's the right side and what's the left side and get the vantage point of the person looking at you. Now, if you tell this lady a joke, notice what happens. She's able to actually move both sides of the face. Now, this would suggest the joke, which is associated with emotional processing and expressions, is now somehow able to control the muscles that previously appeared paralyzed. That's very interesting. This gentleman on the top, he has an injury to his left thalamus. If you do the same thing and you ask him to produce a fake smile, and clearly they've been asking him too much, so he doesn't really smile, he seems to grimace in B here. But the important thing we want to focus on is that both sides of his face, he's able to move them. So he has voluntary control of both sides of his face, even though the thalamus is damaged. And that's what we would expect. However, when you now tell him a joke, notice the right side of his face does not respond, suggesting that now that the emotional centers are involved, those facial expressions are not working, and somehow the thalamus is involved in processing that. That top condition is called emotional facial paresis. The bottom one, which is a lot more common, is called volitional facial paresis. They can't have voluntary movement. So taken together, this does suggest that there are subcortical structures that are involved in the processing of emotion and can actually control the muscles of facial expression. And finally, I want to tell you about Phineas Gage. Now, Phineas Gage is one of the most famous clinical cases in neuroscience. And there are many, many little YouTube videos that talk about Phineas Gage. You can Google them or look up them on YouTube. He was working at a railway, putting down railway tracks. And unfortunately, an explosion took place too soon. He was in the wrong place at the wrong time. And this rod went through his skull. Now, what happened next was pretty amazing. He survived the accident. He was taken to the doctor and the rod was removed. And he essentially seemed fine. But over time, it was observed that he experienced subtle emotional changes and social changes. And scientists have reasoned that the reason these changes took place was because this rod went through parts of his frontal cortex, suggesting that somehow the frontal lobe and the frontal cortex are important for processing these emotional responses. Now, many of the changes that Gage experienced have been wildly exaggerated, but there is enough evidence to suggest that there were some changes that did affect him and did have to do with his social interactions and his motivational and emotional responses. So other evidence to suggest that the brain is involved in the processing of emotions. Now let's go back now to the 1800s, when there's a scientist called Paul Broca who used to study the brain. You may know that name because he was very instrumental in describing Broca's area, which is important for the processing of motor production of speech. And he began to identify a series of subcortical structures, which he called the limbic system. And you can see some of these structures there, the cingulate gyrus, the septum or the septal region, the hippocampus and the parahippocampal gyrus, the orbitofrontal cortex. These structures form like a ring just below the cortex. And that's why he called them the limbic system, because limbic or limbus means border. And so he described them as a set of structures just below the cortex on the border. Let's look at a little video that just talks about that limbic system.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss the limbic system. The word limbic comes from the Latin limbus, which means border, and the limbic system was given this name because its structures lie along a horseshoe-shaped area of cortex that appears to be a border between the cerebral cortex and the subcortical structures of the diencephalon. There are many processes associated with the limbic system, but the system is most frequently linked to emotion. There is no consensus on the structures that are considered a part of the limbic system, and some argue that it is too much of a simplification to consider something as complex as emotion to be handled by one group of brain structures. Regardless, these are some structures that are often included in the limbic system. The amygdala is an almond-shaped collection of nuclei found in the temporal lobe that seems to be especially involved with fearful and anxious emotions. The hippocampus is next to and interconnected with the amygdala. Although it is considered part of the limbic system, the hippocampus is generally associated with memory more so than emotion. The parahippocampal gyrus is an area of cortex that surrounds the hippocampus and also plays a role in memory. The cingulate cortex or cingulate gyrus is found just above the corpus callosum and is involved in various aspects of emotion and memory. The septal nuclei have connections with a number of other limbic structures and are thought to be especially important to pleasure, reward, and reinforcement. The mammillary bodies are two groups of nuclei that are involved in memory and have extensive connections with the amygdala and hippocampus. The fornix is a fiber bundle that carries information from the hippocampus to the mammillary bodies and then on to the thalamus. The hypothalamus controls hormone release via the anterior pituitary and can exert widespread influence over bodily states to maintain homeostasis. While there are other structures that may be included in the limbic system, the structures identified here are some that are commonly considered part of it.
So that's a little video that highlights many of the structures that we now believe are involved in the limbic system. Now, when Broca first described the limbic system, he didn't include all of those structures. But Papez, another scientist, came along in the 1900s, and he began to suggest that these structures are important for processing emotion. And he described something called the Papez circuit, which you can see on your diagram there. And his circuit talked about a flow of information from the cingulate gyrus to the hippocampus, to the mammillary body, to the anterior thalamic nuclei, and back to the cingulate gyrus. And he said that this circuit was important for emotional coloring. So he suggested that when a stimulus was experienced and processed by the cortex, it would then be sent to the limbic system. And as that information circulated or passed through the limbic system, the emotional expression would be given to the stimulus, and then outputs from the limbic system would lead to these emotional expressions. And we would call this, he called this emotional coloring. And part of the reason he came up with this was because we know that damage to many of these structures does lead to problems with emotion. Now, a little while after Papez, another scientist, McLean, said, listen, you might be identifying these subcortical structures, but there's an important structure that I think you've left out that's very vital in emotions, and that's the amygdala. And McLean put the amygdala at the center of this circuit and readjusted the circuit. Part of the reason why McLean did this was because of work taking place on something called Kluver-Bucy syndrome. Now, in Kluver-Bucy syndrome, there's bilateral removal of the medial temporal lobes. And this work was being done on monkeys. The amygdala was removed inside of these studies. And what it was found, when this happened to monkeys, they had an absence of emotional responses, especially responses like fear, rage, and aggressive behaviors. So here in this top picture, you can see a monkey reaching out to grasp a rubber snake. Now, monkeys are usually very fearful of snakes. And so this behavior that we're seeing here in the monkey as it's reaching out to grasp and explore the snake is very unusual and suggests that there's no emotional response of fear. Kluver-Bucy syndrome was also associated with other behaviors like compulsion to focus on tactile and oral stimuli, hypersexuality, as shown in this picture here, and also visual agnosia, the inability to recognize objects visually. This is believed to be due to damage to areas caudal to the amygdala during the process to remove those medial temporal lobe structures. Well, since the time of MacLean, the amygdala has actually become the structure most and best studied with response to emotions. Now, the amygdala, like the basal ganglia, is not really one structure, but it's an almond-shaped mass of neurons. That's actually a collection of nuclei. And you can see some of the nuclei here, the central nucleus, the basal nucleus, the medial nucleus. These structures receive massive sensory input from many different parts of the body and also connect to many of the different parts of the brain that are involved in emotional processing. So if you look at this diagram here, you can see that sensory information comes to the thalamus and then goes to the amygdala. And sensory information also gets to the amygdala from the cortex and from the hippocampus. The amygdala then connects to a lot of different structures that are involved in emotional responses. It can connect to the central gray matters of the brainstem that affect emotional behavior, the lateral hypothalamus that affects autonomic responses, structures that lead to hormonal release like cortisol, and also structures that lead to the startle response in the superior colliculus. And so the amygdala is believed to be an important point where sensory information comes in and emotional responses can be processed and exited via the different structures of the brain. Much work has been gone to try and understand the amygdala, and it has been shown that during emotional activation, the amygdala does function, especially for fear. And so the amygdala has now been particularly associated with the fear response. And you can just see from these studies here that fear responses produce a much larger response in the amygdala than happy responses. And this is true for many negative responses, which seem to have a greater effect on brain processing than more positive responses. And that's believed to be due to evolutionary pressures concerning survival. Now, this is an interesting case study. We looked at an example in animals where persons who had removal of the amygdala developed Kluver-Bucy syndrome. Now, this doesn't occur very often in humans, but there are a few cases where this occurs. And these persons also don't express emotional responses. And one well-studied case is a lady called patient SM. And let's just listen to a little talk about SM.
So SM is one of the most famous case studies in all of affective neuroscience. She's a woman who is currently, I think, in her 40s. And as far as we can tell, the destruction of her amygdala has left her essentially fearless to at least external stimuli. And to test whether SM is in fact fearless, the researchers who worked with her took her to a couple of the most frightening places that they could think of in the local area. So one was an exotic pet store, and they offered her exotic snakes to hold in the store, even though she says she's afraid of snakes. Many people would hesitate to hold a snake right up to their face and touch its tongue and inspect its face really closely, but SM had no problem doing that. They also took her to a place it is at least called the most haunted house in the United States. And they decorated up for Halloween, and they have the staff dressing up as monsters and things to try to scare people. And the researchers in SM went through with just a regular group. And all the other people who were in the group, as soon as they were in this haunted house that is apparently very spooky, were, huddling together, and they were really slow to go around corners. And SM was taking the lead and saying, "Come on, guys, follow me." And she would be the first around the corners. And when the monsters jumped out and tried to scare her, apparently she would scare them sometimes because she had no fear response to them, which they were not expecting. And so what was really interesting about this is that SM is not emotionless. She experienced a lot of curiosity and excitement, which we think are governed by other regions of the brain predominantly. But she just doesn't seem to have any normal anticipatory fear response. But her lack of a fear response gets her in trouble. So she apparently walks home in the evenings through sort of a vacant lot. And she was mugged there at one point, and I think at knife point. And most people, when something scary like that happens to you and you are at risk of getting hurt, you have a normal fear response when you're approaching that same place that would send you avoiding it. I mean, that's what the fear response is all about, avoid things that might hurt you. And she doesn't have that response, so she continues to take that same route home every night by herself. Yeah.
So that's just an interesting description about patient SM, who doesn't seem to have the fear response. And you saw some of the things that happened to her. Interestingly, we all have something called personal space. And studies have shown that the average personal space for persons is a little bit over a meter. But SM, her personal space is much smaller. That's just interesting, an interesting side effect of what the amygdala might be involved in. Other work with the amygdala has shown that if you actually stimulate it, animals show increased vigilance and attention. Again, explaining the importance of the amygdala, in processing fear and helping with motivation and drives. And finally, a lot of work in trying to understand the amygdala has focused on fear conditioning and helping us to understand how a brain structure can be involved in learning and motivation, and how that also is linked to emotions. So let's look at this other little video that talks about
The amygdala, meaning almond-shaped, is a collection of nuclei within the brain. It is a paired structure with one in each hemisphere. The amygdala is thought to play a role in a number of different functions, but it is most commonly implicated in the control of fear. The amygdala is located within the medial temporal lobe and is composed of a number of different nuclei. Some of the main nuclei are the lateral nucleus, the basal nucleus, the central lateral nucleus, and the central medial nucleus. There are also groups of intercalated cells dispersed within the fiber tract of the nuclei. Two important groups of intercalated cells are the dorsal intercalated cells and the ventral intercalated cells. The lateral nucleus is the primary input nucleus of the amygdala. It receives inputs from the thalamus and the cortex, which provide it with information about the sensory stimuli the animal is experiencing. The primary output nuclei of the amygdala is the central medial nucleus. This projects to a number of different structures. It projects to the paraventricular nucleus of the hypothalamus and triggers the release of the stress hormone cortisol. It also projects to the lateral hypothalamus, stimulating the autonomic nervous system. And it also projects to the periaqueductal grey matter, which in mice causes fear behaviors such as freezing. By coordinating these downstream targets, the amygdala is able to produce many of the physiological changes associated with feelings of fear, including increased heart rate, sweating, and dilation of the pupils. One of the best studied functions of the amygdala is that of fear conditioning. Put simply, this is where an animal learns to fear something. In a typical fear conditioning experiment, a mouse is given an unconditioned stimulus, which is inherently negative, such as a painful foot shock, which causes a natural, unconditioned response, in this case, the expression of fear. The experiment also uses a neutral stimulus, such as a sound, which on its own produces no fear. After a number of trials, when the neutral and unconditioned stimuli are presented together, the animal learns the association between the sound and the shock. The neutral stimulus of the sound is then able to cause the fear behavior itself, without the shock. As the process of learning in this way is known as classical conditioning, The stimulus is then referred to as a conditioned stimulus, and the response referred to as a conditioned response. Related to fear conditioning is the concept of fear extinction. Extinction occurs when the conditioned stimulus of the tone is presented repeatedly without the unconditioned stimulus of the foot shock. The animal gradually learns that the sound no longer predicts a shock, the association is broken, and the fear behavior is no longer displayed. The amygdala is thought to be the primary area of the brain responsible for fear conditioning, and the classic model of how it does this is as follows. The unconditioned stimulus of the foot shock travels through the spinal cord to the thalamus and the cortex, which then both project to the lateral nucleus of the amygdala. The synaptic inputs from the unconditioned stimulus of the shock are strong enough to excite the lateral amygdala neurons, which in turn results in the activation of neurons in the central medial nucleus and produces a fear response. The neutral stimulus of the sound also flows through the thalamus to the auditory cortex. The thalamus and cortex then also project to the lateral nucleus of the amygdala. However, the inputs from the neutral stimulus alone are not strong enough to cause the lateral amygdala neurons to depolarize, so the central amygdala neurons remain unstimulated and no fear is displayed. However, when the neurons encoding the shock and the neurons encoding the sound fire together, synaptic plasticity occurs, like we've seen in the previous videos. This strengthens the synapse between the incoming neurons carrying information about the neutral stimulus of the sound and the lateral amygdala neurons, until eventually the synapse is strong enough to allow them to stimulate the lateral amygdala neurons on their own without the help of the neurons encoding the unconditioned stimulus of the shock. The lateral nucleus of the amygdala then excites the central medial nucleus and produces feelings of fear. The neutral stimulus then becomes a conditioned stimulus and is able to produce the conditioned response of fear without the foot shock. After fear conditioning, mice not only express fear with the conditioned stimulus, but also when returned to the chamber in which the experiment took place. The mice have learnt not only to fear the stimulus, but also the context surrounding it as well. This is known as contextual fear conditioning. One of the primary areas responsible for this is the hippocampus, which, as we have seen in previous videos, plays an important role in storing episodic memories. Neurons from CA1 and the subiculum areas of the hippocampus project to the basal nucleus of the amygdala, and through this are able to stimulate the central medial nucleus and allow contextual cues to produce fear. In conclusion, we can see that the amygdala is a cluster of nuclei within the brain, which produces many of the physiological aspects of fear. The amygdala also plays a key role in helping us learn what to fear, and the best studied example of this is fear conditioning. In fear conditioning, synaptic plasticity allows a weaker sensory stimulus to trigger a fear response when paired with an unpleasant stimulus. The amygdala also interacts with other brain areas, such as the hippocampus, to allow context to affect our expression of fear.
So I thought that was a very useful video because it summarized many things. It summarized the structure of the amygdala, the anatomy. It summarized the connections of the amygdala and how those connections are important for receiving sensory information and outputting via structures that affect the emotional response. And then it went on to talk to you about how by studying the structures of the amygdala, we've been able to understand the processing of fear. And today, much work continues to take place looking at the role of the amygdala and fear. One of the things you would have seen is that sensory information is able to reach the amygdala very quickly. And this is believed to be one of the reasons why we have the amygdala and why we have invert motion. You can see that the standard route that information goes through to reach the amygdala is by the thalamus and the cortex. But there are also other what are called emergency routes, where it can go directly from the thalamus to the amygdala. And these routes are obviously a lot quicker, and therefore they result in a very rapid response in situations that we've learned are fearful, even before we're even consciously aware. For example, seeing something that's dangerous. We also saw in the video that the hippocampus and from the limbic system structures is very much connected to the amygdala. And we saw that the hippocampus is important for contextual learning and fair learning. And that's because there are parts of the hippocampus that's involved in spatial awareness and spatial memory. And then there are also parts of the hippocampus that connect to the amygdala and can transfer that information and use it for emotional processing. And this is why there's such a strong relationship between emotions and memory, because of the connections between the amygdala and the hippocampus. Okay, let me just conclude by not so much focusing now on brain structures. We've seen many brain structures that seem to be involved in processing emotions. We've seen the role of the frontal cortex, the orbitofrontal and the ventromedial cortex, which were damaged in the case of Phineas Gage. We've seen the limbic system and the different structures involved in the limbic system, like the hypothalamus, the amygdala, the hippocampus, all are involved in emotional processing. But behind all of these structures, or within all of these structures, are neurons that are using different neurotransmitters. One of the neurotransmitters that seems to be particularly associated with emotions is serotonin. Now, serotonin neurons are associated with the reticular formation, and there are nuclei within the brainstem, the raphe nucleus especially, that project down to the spinal cord and throughout the central nervous system, the brain, as seen in this diagram. And you can see that these serotonergic projections go to many emotional processing centers of the brain. It has been shown that if you reduce the amount of serotonin in rodents, it can lead to aggression. And if you do that by reducing its turnover or reducing its synthesis, the results are the same. It has been shown that if you give persons a diet that's low in tryptophan, the precursor of serotonin, it can lead to negative mood bias. And that this can even lead to changes within the connections of the brain that resemble what happens during depression. Now, this has led, along with other studies, to the development of drugs that are able to boost the amount of serotonin in the brain. And these drugs are classically known as selective serotonin reuptake inhibitors. They block the pumps that return serotonin to the presynaptic neurons. The classic example of this is a drug, trade name Prozac, fluoxetine. This is often used to treat depression, and it's shown that lower doses can help with mood conditions. And you can see here, it can even help with the processing of emotions. So here you see somebody in the study here who's been exposed to fearful faces. And then we're looking at the activation of the amygdala. Persons who are given a placebo had good activation of the amygdala. But persons who were given an SSRI and they were boosting their amount of serotonin, they had much less activation of the amygdala. It's also been shown that SSRIs can improve confidence and cooperative behaviors. And some people now describe serotonin as a pro-social hormone. It produces pro-social interaction by reducing some of these negative emotional experiences. So in seeking to understand emotions, we both have to look at the structures involved, the circuits involved, and the neurotransmitters involved. And there's tremendous efforts to understand this, both for normal physiology and pathological conditions in mental health disorders. So that brings us to the end of our tour of the limbic system and emotions. In summary, we've tried to define emotions and look at the three components of emotions: the subjective feelings, the physiological arousal, and the behavioral or expressive component. We've looked at some of the psychological theories behind emotions and traced their evolution from the James Lange theory to the Cannon-Bard theory to the two-factor model. We've looked at evidence to support the fact that there is neurobiology responsible for emotions, the fact that across all cultures there are basic emotions, and the fact that in clinical conditions it can affect emotional expression. And then finally, we spent some time looking at the limbic system and the different structures in the limbic system, and how when they are damaged, they can affect emotional processing, and trying to see what is their role in emotions. Obviously, a lot more work to be done, but an area of neuroscience that is rapidly expanding. Until next time, take care.